Okay, so welcome everyone. This is our first BiteSizeResearch webinar. As some of you may know, we usually hold a conference, the Bone Cancer Conference, every year at the Bone Cancer Research Trust. And unfortunately, it was originally due to take place next month, but we were unable to go ahead with it because obviously the current situation because of COVID-19. So we thought it would be a great idea to hold these weekly webinars so that you can learn more about our research and potentially get some updates that you would have potentially got at that conference, but from obviously the comfort of your own homes. So I'm joined by my colleague, Dr. Zoe Davison, and my other colleague, Dr. Viqui Vinader. And my job is to kind of host you all and keep on top of any questions you might have and try and make sure we stick to time. So we've got 44 people on. If I could just ask everybody to keep on mute, then obviously we don't have any background noise and hopefully we won't have too many glitches as we go along. So just to introduce ourselves, and so I'm the Head of Fundraising and Communications at the Bone Cancer Research Trust, Matt Coppell-Shaw, and like I said, my colleague Zoe and my other colleague Vicky are joining me, who are part of our research information and support team. They're going to be leading today and giving you a talk, and then we will have questions at the end. So we've got about 5 minutes now just to allow people to join. There's just a couple more still wanting to join. And then we'll have about 30 minutes, 35 minutes of the talk that Vicky and Zoe have prepared. And then there'll be 15 to 20 minutes for questions at the end. So you should all have access to the chat function. And what I would just suggest is if you could keep that up, if you have any questions, if you just pop your question into there, if you could type it in, then at the end I'll try and make sure we answer as many of them as we possibly can do. So just one thing to say, the webinar is very much focused on research. It isn't about personal circumstances or situations to do with treatment. If you do have any questions related to that, then our support and information team are available and we can give you details of that separately. But if you have any questions related to research, please do fire them away. We have a few that were asked in advance, so we've got those already. But like I said, if you could just put your questions into the chat function, I'll try and keep on top of them throughout. So Zoe, over to you.
Okay, so my name's Zoe Davison. I'm the Head of Research Information and Support at Bone Cancer Research Trust. So I'm going to talk to you first. So the first part of our webinar will be focused on how BCRT started and what progress we've made to date. And then I'll hand over to Vicky, who will then talk to you about some of the kind of fascinating emerging trends that are coming up now in cancer research and how we are facilitating those in primary bone cancer research.
Vicky, do you want to introduce yourself?
Yes, hi. So I'm Viqui Vinader and I'm the research manager, and I will be following on from Zoe's presentation. So it's a little bit of a superficial view, so please bear with me. But if you, I wanted to stress that if you want any further detail of any of the points that we mentioned, please feel free to contact me or Zoe, and then I'm very happy to talk to you in more detail about any of the projects or any of the things that we talked about.
Okay. Fab. So hopefully technology is not going to fail us now, and I'm about to share a presentation with you all. And I promise you, we've not taken our lead from the daily government briefings, but to try and limit any technological issues, I'm going to be forwarding the slides. So Zoe and Vicky are going to be instructing me to move us on. So, okay, so you should all be able to see the presentation now. Zoe?
Yes. Okay. So as Matt alluded to, this is the first webinar in a series, and the first one really is to just give you a flavour of some of the really exciting projects that we funded and we are beginning to fund. And so we kind of touch on some of those aspects so that it'll give you a kind of insight into what's coming up further in the series. Next slide, please. So in this webinar, we will look at why and how Bone Cancer Research Trust was formed, where we are today, some of the novel and exciting areas of research that are coming up in kind of the broader cancer research area, and how they relate to primary bone cancer research, and then a little bit about how BCIT is responding to these new cutting edge areas with our research projects. And then there'll be some time for some questions and answers. Next slide, please. So firstly, I thought I'd explain how and why Bone Cancer Research Trust was formed for those of you who don't know. So back in 2004, there was no national charity dedicated to primary bone cancer or bone sarcomas. And a group of families who had lost children and young people to bone sarcoma were brought together by an oncologist who was based in Leeds called Professor Ian Lewis. So he'd noticed that they were all doing their independent fundraising because they were all quite upset by the lack of information available to primary bone cancer patients and the support that they could receive, and actually quite shocked at the lack of research that was being done in this area. So after a few meetings, some of the families decided to work together and pool their funding, and in 2006 Bone Cancer Research Trust was registered as a charity. And the two images that you can see, the pictures, is a bit of an ode to our previous logo. So the two gingerbread men represent the two age groups that primary bone cancer affects, so young people and older people. Next slide, please. So in the early days, the charity's focus really was on stimulating research. So you can see on the graph, so this is quite a busy graph. So this graph shows the number of publications. So publications are in scientific journals that are a result of knowledge that's accumulated through research projects. And in the early days, you can see that there's very, very few publications in primary bone cancer in the UK. And then after 2006, when Bone Cancer Research Trust was formed, it's nice to see that publications go up. So our knowledge is increasing since Bone Cancer Research Trust was registered. And I would love to take credit for all of those publications, and that BCIT was involved in all of them. But it's around 25% of publications in the UK have come about partially due to or fully due to Bone Cancer Research Trust funding. And what we want to do is fund research, but also fund pilot research that then can lead on to more substantive research funded by other organisations, just to acquire that knowledge and stimulate that research. And to date, there's been 71 peer-reviewed scientific publications that have resulted from Bone Cancer Research Trust funding. Next slide, please. So stimulating research is important in the earlier years. However, it's important to review this funding periodically. And as time went on, we wanted to move away from just stimulating any kind of research to actually focusing on what is important to our stakeholders and what's going to make the real difference to our patients. So in 2015, we undertook a piece of stakeholder research, and we surveyed 321 patients, family members and friends, healthcare professionals and researchers. And it looked at three main things. So what a diagnosis of primary bone cancer meant and its impact on both the patient and their family and wider circle of friends. And also where there might be gaps in their care or support. Also what Bone Cancer Research Trust means to our stakeholders, and finally where we should prioritise our funding. And the results of that stakeholder research were that patients overwhelmingly felt that our research spend should focus on the cure. That was the most important, so finding new treatments, better treatments in order to improve outcomes and in survival for patients. and also the causes of primary bone cancer. The survey also showed that our researchers felt that Bone Cancer Research Trust should review the types of grants offered and really promote collaborative research and translational research that kind of move laboratory research towards the clinic. And there was also a huge emphasis on patient samples in research and overcoming the obstacles in using and collecting those samples. Next slide, please. So following on from the stakeholder research, we went out to our research and clinical community to see really where their focus was and where they felt our funding should be prioritised in order to make the best impact and the most use of limited funding. So we spoke to our researchers and our clinicians, and we worked with the National Cancer Research Institute Sarcoma Research Group and also the bone subgroup of that group to understand where the most urgent needs were and where we should prioritise our funding. Next slide, please. So in response to these two pieces of research, we launched our new strategy, so the biggest ever commitment to primary bone cancer, in July of 2017, and it covers our strategic aims from 2017 to 2022 across all the pillars of our charity, so research, information, support and awareness. As part of this strategy, it also details our grant programme that runs from 2017 to 2022. And for the first time, it gave researchers in our community a clear picture of where funding calls would come and what was going to be available in each year, so that they could forward plan their research and know when funding was available. We also introduced two new types of grants for our new grant programme. So we introduced our infrastructure grants, which are focused on sample collection, and also our clinical and translational grants. And the focus of these are to take early and laboratory science through to more clinical science and more patient-facing research, with the hopes of changing clinical practice. Next slide, please. So one of the main areas that we needed to address was the availability of patient samples for use in research. Patient samples really are the best tool and the best way of understanding what goes wrong, what causes primary bone cancer, and in turn how to treat these. And we know from studying patient samples that each cancer is very different, and we need to look at them as different and different entities. So in 2016, bone sarcomas became eligible for analysis in the 100,000 Genomes project, which was a government initiative. And this really actually focused our mind and made it really, really important that we had grants in place to support this activity. These samples are going to be analysed for free so that we got a really good understanding of the genetics of bone sarcomas. And without our sample collection grants, this wouldn't have happened. The samples wouldn't have made it into this project. And so in 2017, we launched these new sample collection grants, and they give small pockets of money to the 5 surgical centres in England to use in how they see fit, either to employ staff to allow for the collection of samples, or for equipment, or for different things that are needed to undertake this activity. So the grants themselves don't actually fund research directly, but what they do allow for is the collection of samples to be used in different research projects, both funded by Bone Cancer Research Trust and funded by other organisations. And importantly, they allow for the collection of bone sarcomas and soft tissue sarcomas. We didn't want to put any restrictions on what samples could be collected as part of these. So since they were launched, we've collected 2,295 samples. They've been used in 16 different research projects that we know of, and others have been banked for future use. And they've actually gone on to support 2 quite important projects that have been launched by Sarcoma UK. And so it just emphasises the importance of what we can achieve when funding is aligned and between charities. Next slide, please. So while working with the NCRI Sarcoma Research Group, it became really apparent that there was quite an unmet need and challenge in osteosarcoma research, particularly in trials and biological studies. There hadn't been a phase 3 trial in osteosarcoma since Euromos, which is over 10 years now, and there was limited early phase clinical trials and very limited biological studies. So in 2018, Bone Cancer Research funded our first clinical translational grant, ICONIC, and it really is an exceptional example of how collaboration can work and how important it is. It's brought together 24 NHS trusts across 35 sites, and it is the biggest collaboration in osteosarcoma research in the UK. So it aims to recruit every newly diagnosed osteosarcoma patient with osteosarcoma in the UK, and irrespective of age, and it will develop the UK's first ever patient registry, which will bring together both clinical data and their tissue samples so that we can map what treatment a patient gets, how they respond to the treatment, and actually what's going on in their tumour, so that we can try and work towards developing stratified and personalised treatment for our patients, which is quite elusive in bone sarcomas at the minute. So the aim is to enrol 200 patients over 3 years to build a platform that when new treatments are available, they'll come on and be added to this clinical trial. So far, 26 patients across 15 hospitals have been enrolled onto ICONIC, and we have a wide age range. We have patients under 10 and we have patients over 60. So we're hoping that this project will give us some really good results. As part of our osteosarcoma work, we're really keen to work closely with an American group on harmonising data that we get from ICONIC. So you may have heard of Count Me In. So there's an osteosarcoma component of Count Me In that's just been launched, and we are hoping that we can harmonise the data from that and from ICONIC so that we get some really, really good data and some important results from these. Next slide, please. So another thing that came out of our stakeholder research, and it's very important to Bone Cancer Research Trust, is collaborating with other organisations. For a number of years, we've worked very closely with Ewing Sarcoma Research Trust to fund vital research into Ewing sarcoma, and we have a webinar confirmed later in the series from Professor Graham Cook and Tyler Barr, who will go and explain some of our research in Ewing sarcoma, and we're hoping to organise another webinar later in the series on some more research that we've funded in Ewing sarcoma. So if you're interested in this area of work, please do tune into some of the other webinars. More recently, we've worked with children with cancer to facilitate osteosarcoma research, and in 2019 we held our first international osteosarcoma research symposium, and this brought together experts from America, from Europe, and obviously the UK, and it was a really inspiring day. We talked about where we are with osteosarcoma research, where the gaps and challenges are, and how we can work together to overcome those. We're also working with Clive and Sylvia Richards charity to fund a programme of research focused on the genetics of primary bone cancer and the development of new treatments. We are also working with charities and organisations to really understand what's happening kind of globally in the primary bone cancer setting, so that we can align our funding strategically so that we're all building into a bigger picture rather than working in silos and funding work separately. We really believe that together we can achieve more and more impact for our patients. Next slide, please. So where are we today? We've come quite a long way since 2006 when the charity was registered, and today we're the leading charity dedicated to fighting primary bone cancer. We offer hope to all patients affected by the disease. Our vision is a world where primary bone cancer is cured for all patients, and our mission is to save lives and improve outcomes through our 4 pillars of research, information, awareness and support. We're a small but very dedicated and passionate team, and unlike a lot of charities, we're based in the north of England in Leeds, which is in West Yorkshire. And to date, we have funded 68 principal investigators, 95 co-applicants and 75 collaborators in 83 unique research projects. And our funding has totaled nearly 4 million to date. Next slide, please. But there's still a lot to do. So research is like a jigsaw puzzle, and as we have funded projects, pieces of the jigsaw puzzle are coming together and we're starting to see some of the picture, but there's lots of pieces missing. So we've funded some really successful projects on looking at surgical margins and surgical techniques. We've funded some really important work looking at protocols for new radiotherapy techniques, including proton. We facilitated patient sample collection, and we've looked at different models, so 3D cellular models of bone sarcomas, but there's still a way to go. So I'm going to hand over to my colleague, Viqui, now, who will talk about some of the missing jigsaw pieces and how we're trying to put them in place.
Thanks, Zoe. Matt, can I have the next slide, please? So following on from Zoe's introduction, what we're going to do in the second part of the presentation is to take you a little bit through some novel trends in cancer research that are now making progress into primary bone cancer research. So Zoe and I have had a thought about this, and we selected 4 research areas. Of course, this is not everything, but these are examples that we think really show the changing landscape of bone cancer research. The first one is genetic modifications. These are responsible and really drive the cancer progression. So we want to look at those in order to identify new therapeutic targets, also to understand how cancer evolves. And the second one would be immunotherapy as a potential treatment option for primary bone cancer patients. The third, biomarkers that will allow us to get a better diagnosis and also monitor of the disease. And finally, and very briefly though, we'll look at some potential new laboratory models to facilitate primary bone cancer research. So can I have the next slide, please, Matt? So we start with the genes. Now we know the cancer is caused by changes in DNA. Those changes alter the cell behavior and then they result in uncontrolled growth and malignancy. But why is the connection? The reason why genes have an effect on growth and malignancy is because genes contain the information for the cells to make proteins, and proteins actually control cell growth. So DNA changes instruct cells to produce abnormal, and that could be more or less, of proteins that have altered shape. And when proteins change the shape, they may not work as they should do. And these genetic alterations may intensify or eliminate altogether the function of the protein, and that could cause cells to go into dividing uncontrollably. Can I have the next slide, please? So in this context, we can talk about cancer genetics or cancer genomics. The first one refers to the genes that we inherit, and genetic tests can help us identify a person's risk of developing a particular cancer. As a reminder, you know the DNA contains 4 basic building blocks. So there's A, C, G, and T. And it's the order, what we call the sequence of these 4 letters, what defines how they're arranged, what defines our genetic information, what we call our genome. So cancer genomics studies the differences between the DNA sequence, the order of those letters, between tumor and normal cells. And identifying the difference is fundamental and is really important because, of course, it facilitates the diagnosis, allows prognosis, and guides the treatment for each patient differently. Next slide, please. So clearly, since we need to see the differences, we need samples of tumor and samples of healthy bone. And obviously for those, we need to obtain samples. That goes back to our infrastructure grants. So I've done a little scheme here for you, which you might think it looks a little complex, but even within that, it's a simplified scheme. But you can see the cancer genome sequencing, the discovery of disordered letters and the difference between tumor and normal is a complex process. There's no two ways about it. It does also generate a humongous amount of data, and it is probably the analysis and the interpretation of the data that is the most challenging. So our infrastructure grant have guaranteed the collection of samples and of course have been pivotal in including primary bone cancer patients in the 100,000 Genome Project. They've also allowed our scientists, our researchers to participate in something which is called the International Cancer Genome Consortium. That was started in 2007, and it is simply a collaboration, a massive group of scientists to share data in terms of genomics throughout the world. Can I have this next slide, please? So genomics identify individual changes in genes that drive the cancer behavior. However, we do know that the changes in tumors occur over time. And this is because more genes continue to mutate. And in fact, we know that patients may develop resistance to a certain treatment that originally was working. Here on the right, you see a little cartoon of Darwin holding his famous evolutionary tree of life. And that is really nicely mirrored on the left by a cell that undergoes a change and becomes a cancerous change, and there goes further mutations and so on and further on. So it almost mimics the evolutionary tree. So scientists are now using Darwinian principles to understand the genetic complexity of advancing tumors, and particularly metastasized tumors, and try to use this knowledge to devise approaches that manage to control them. Next slide, please. So one of the ways that cancer genomics can help is to find novel targeted therapies. An example is the work that Professor Susan Birchall at University of Leeds has been carried on. She has a long-standing interest in how, by understanding the genetics and also the biological characteristics of human sarcoma cells, we will be able to find new targets for treatment. She has able to identify genes that are responsible for the production of certain proteins. An example is neurexin-1. And this particular protein is especially abundant in a subpopulation of cells, human sarcoma cells, which are resistant to therapy and cell renewing. They're called cancer stem-like cells. Now, to identify proteins that specifically are present in that population of cells is fundamental because it gives us a handle, something that we have in those cells and they're not in other cells, that gives us a handle to find them and treat them. It also allows for us to identify these patients that have these proteins earlier on in the treatment, in the diagnosis, to inform their treatment. Next slide, please. We move on now to our second area, which is immunotherapy. Now, we know that immunotherapy in general uses our immune system to fight disease, cancer in particular, and it has received a lot of attention in recent years. Some of you might be interested in and might have heard of something called immune checkpoint inhibitors. So I'm just going to spend a minute just describing them very briefly. So cancer cells are different. They have changed and they're therefore foreign to our bodies. So the immune system should in principle be able to destroy them. However, they often disguise and are able to avoid this. So one of the mechanisms by which cancer cells are able to avoid destruction by the immune cells is by stepping on breaks that are present on the surface of immune cells. We call these breaks checkpoints. These checkpoints are there, they're a normal part of the immune cells, and they're there to prevent them from overreacting, so it's normal. The only thing is that cancer cells take advantage of them and use them to avoid being destroyed. Can I have the next slide, please? So we're doing a little cartoon to illustrate this. So here on the top part of the skin on the left, we have an orange blobby cell, which is a cancer cell. And you can see that there might be a protein I highlighted in blue on the surface, and that protein allows an immune cell in yellow to find the cancer cell and recognize it for destruction. So if you can find it, it's destroyed. But we just said that cancer cells avoid immune destruction by acting on these breaks, these checkpoints. So we highlighted the breakpoint as a gray arrow. So if you go to the second part of this, yeah, there you go. So cancer cells almost like grab those checkpoints, and that inhibits the immune cells from being able to destroy the cancer cell. So what immune checkpoint inhibitors are, highlighted in the middle of the slide with that little Y in purple, is that they disrupt that interaction, and therefore they free the immune cells and are able to act on the cancer cell and destroy them. So this approach was originally applied to mostly for late stage lung cancer and also melanoma, but it's now been extended to quite a variety of cancers. And in fact, several clinical trials are now looking at the application of immune checkpoint inhibitors for primary bone cancer, mostly osteosarcoma, but not only. If you are interested, you can contact me and I can give you some more information about that. And next slide, please. Another approach which is really interesting, and maybe if you hear virus now, you think not viruses, but these are a really different type of viruses. They are engineered viruses. They're called oncolytic viruses that are designed to affect only cancer cells. So when they encounter a normal cell, they're not able to replicate inside the cells and destroy it. However, when they find a cancer cell, they get inside, they replicate, they multiply, and then they're able to destroy. The cancer cell ends up being burst. So as a result of the burst, there's two things that happen. One, that the viruses are liberated within the environment, and then they might find another neighboring cancer cell, do the same and destroy it. But there also would be the release of antigens. And then the antigens would act like beacons and then call up other immune cells that will come up and destroy them. Next slide, please. So it's a really interesting idea, but it seems a little bit far-fetched, but it actually is a reality. There's already approved drugs, approved oncolytic viruses that have been used for treatment of cancer. The first one was called TVEC, and it's been used for the treatment of melanoma. And currently we have a PhD studentship at the University of Leeds. So Tyler Barr, who is a PhD student, she's working with Professor Graham Cook and Dr. Fiona Errington-Mays, and she's looking at the possibility of studying oncolytic viruses to treat Ewing sarcoma. And they will be participating in a coming up seminar. So if you're interested in this topic, please attend that. Next slide, please. The last approach I wanted to highlight in terms of immunotherapy is the use of tumor-associated macrophages. Now, macrophages are big white blood cells, which job is to mop up debris. They engulf and eat up anything that's unwanted. This happened to accumulate around tumors, particularly osteosarcoma, and in that context, we know them as tumor-associated macrophages. So tumor-associated macrophages actually help cancer cells avoid destruction by the immune system by releasing, making some of the proteins. We currently have a research project which is taking place at King's College London and is led by Professor Agi Grigoriadis. He's looking at one particular protein, which is called heme oxygenase 1, which is produced by tumor-associated macrophages. In other cancers, for example, breast cancer, this protein has been demonstrated to avoid the destruction of cancer cells by the immune system. Next slide, please. So what the team has managed to do is to show that tumor-associated macrophages in osteosarcoma also produce this protein. Now, we can't stop the protein being produced, but what we can do is to stop it from functioning. So we want to inhibit it. So drugs that inactivate heme oxygenase 1 actually exist, and we can take advantage of them. The beauty of it is that they are used for some other indications, for example, neonatal jaundice. So they are very safe because they're used for babies. So the project is currently looking at investigating if, either with or without chemotherapy, drugs that act on heme oxygenase 1 would be able to reduce osteosarcoma growth and metastasis. Next slide, please. The third area of research we wanted to highlight this afternoon is biomarkers. Now, biomarker in general and cancer specifically is something that can highlight the presence of cancer. And we can use them for many things, really. We can use them to estimate the risk of the disease, for screening, for differentiating whether we have a malignant or benign tumor, or defining the type of tumor. And therefore, obviously, defining prognosis. Also to monitor the disease and the response to treatment as well. Can I have the next slide, please? So one example we wanted to bring out this afternoon is the use of circulating tumor cells as biomarkers. So if you look at the right-hand side of the slide here, you see a tumor mass, and cancer cells can detach themselves from their original tumor mass. They can kind of burrow through the environment, find a local blood vessel, and enter the circulation. Once in the circulation, they can then go anywhere in the body, exit through another blood vessel, find a local environment, and grow another tumor, a metastatic deposit. And these traveling cancer cells in the circulation is what we call circulating tumor cells. They're in the bloodstream, so therefore we can obtain them with a blood sample. The difficulty is that you see there will be very few of them compared to all the other cells in the circulation. There's white blood cells, red blood cells, all sorts of things. So it is tricky, but several techniques and instruments are now available to separate them, characterize them, and actually multiply them in the lab so they can then be studied. Next slide, please. So we have it sponsored Professor Dominic Heyman at the University of Sheffield and Kenny Rankin in Newcastle to isolate, characterize, and investigate osteosarcoma circulating tumor cells. And the protocols that they have developed are now part of ICONIC, which was mentioned earlier by Zoe, and is a landmark observational study for osteosarcoma. Also in the circulation, in the blood, we can find other things that the tumors release. An example we just mentioned, circulating tumor cells. We can find circulating tumor DNA. So tumors release bits of DNA into the blood, and we can measure and detect them through blood samples. So they are obviously non-invasive tests because it's just a blood sample. So we can use them to assess complete or incomplete removal of the tumor. And also we can use them to assess response to treatment or hopefully or not relapse. Next slide, please. So we have two projects that are investigating the potential of circulating tumor DNA as biomarkers. The first, they're both at UCL, and the first is led by Professor Adrienne Flanagan. She's identified a very promising biomarker for chondrosarcoma in this case, which is a mutated gene called IDH1. So through a blood test, you can detect IDH1 levels in chondrosarcoma patients, which are very high before the surgery. And after surgery, those levels drop because the tumor is not there anymore to release the DNA into the circulation. Interesting, the IDH biomarker can only be used to predict the grade of the tumor. So it's really interesting. We have another project, also at UCL, which is looking at a different type of biomarker, also in the circulated tumor DNA, which is a DNA methylation marker, in this case for chordoma and osteosarcoma. And again, as before, they could be used to assess if the tumor has been completely removed by surgery or not, hopefully detect early relapse or response to treatment. Next slide, please. So this brings us to our very last area, which is the need for better laboratory models. The reason for that is because we know that each cancer is different. Even within the same type of primary bone cancer, each tumor would be different, and each patient would respond probably different to another. So one of the main drawbacks in primary bone cancer research has been really the lack of reliable laboratory tools that truly represent the complexity of the tumors. So we need efficient models. We put there a picture of an avatar just to exemplify what we mean. So something that really represents and behave similarly to what the patient's tumor is originally in the body. Can I have the next slide, please? So here it brings it to mention the very last project that we have funded, which is now beginning to cross the barrier between cancer biology and bioengineering. It's a PhD studentship that will look at tissue bioengineering to propagate human osteosarcoma cancers, just like they are in the body, but are actually outside the body. The project is a collaboration between Professor Sylvie Mitnard and Wen Yu Song, and it has two main objectives. One is to find a reliable tool for drug testing. The second, to understand how osteosarcoma cancers begin. And this project is initially focused on osteosarcoma, but if it's successful, we can then extend it to other types of primary bone cancer, which is super exciting. Next slide, please. So this takes to the end of the presentation. We know it's been like a little bit of superficial and a bit fast. However, what we think that all these approaches have in common, and in fact all the other projects that we sponsored over the years, is that they aim to achieve more efficient and personalized treatments for primary bone cancer patients. And I would encourage you to attend other seminars. So we have a list of confirms, but there will be more, so keep an eye. And so for more confirmed dates for other very exciting topics. So after that, thank you very much. And we're very happy, both Zoe and I, we're very happy to answer any questions you have.
Okay, back in the room. Thank you both for that. I'm just going to bring up the chat, which looks like we've had a few discussions on, which is really great. What I'll do first, I'll just turn to the questions that we've had sent to us in advance. Answer those and get those out of the way. So a lady called Caroline Fernandez, I apologise if I've not said that right, has asked, she is an osteosarcoma survivor, nearly 20 years clear, and she's asked, would donating her body to medical research be beneficial to the cause? She's hopeful she will live for at least another 50 years, but would that be beneficial? Vicky?
Yeah, so I do hope that, no, you will believe for a long time, 100%. However, there is an initiative, and what we thought is that I could send you the information if you're interested, or anyone who is interested. There is something called the PEACE study, which stands for Posthumous Tissue Donation in Cancer. I can send you information if you're interested, and of course it will be interesting because it goes back to cancer evolution. If you could find how tumors started and how tumors end, there would be different. And of course, but I'm very happy to send you information if you want on the PEACE studies, if anyone who is interested.
Okay, another lady, Sarah Moses, asks, I'm a fundraiser for the charity. I would like to know how the work I do translates into the work you do. Zoe, do you want to do that one? You're on mute. There you go. Oh, go on, try again. There we go. We'll get you off mute in a moment. You off? There you go.
Fabulous. So money raised by supporters, were obviously very, very grateful. So the money goes to support our 4 pillars, and we have in our strategy where the money goes to support these 4 pillars. So last year we launched our new support and information service, but the emphasis of money donated goes to our research programme. And in our strategy, you'll find our grant program, which lists the funding calls that we are committed to. But that's kind of a framework, and if we secure more funding, the plans are to build on that framework and add more funding calls to that. And over the last couple of years, we've added an extra PhD studentship and an extra Explorer grant because our fundraising has exceeded what we expected, and so we wanted to put it to the best use in our research programme.
Well, thank you. Okay, I think this is a question maybe towards both of you. Philip Dixon has asked, have Ewing sarcoma survival rates improved since he was a patient in Old Middlesex Hospital in 1994? So have there been survival rate improvements since 1994? Viqui?
So obviously up to 2000 and including the 1990s, survival was worse. So from that time approximately, the survival rates for Ewings remain, say for example, the 5-year survival rates remain between 66 and 68%. The problem, we would say, is that when we look at overall survival rates, they are averaging everything. So we can see that they are, and we know for sure that they are improvement in survival, but when all the data is averaged, the detail gets lost. So when we say there is a uniform ratio of 66%, it doesn't include the fact that the different morphology, the different anatomy, whether it is in the leg or whatever. So we are currently working, hopefully, with Public Health England, try to get more detailed analysis of the data, and I'm pretty sure that we will see increases in survival, because it is the average of the data that makes it look uniform and not changing, rather than the reality. Zoe, I think you have some comments about this as well.
Yeah, so I'm sure some of you know that there's been a trial recently called Euro Ewings 2012. It's just closed. And it's been a successful trial actually, and we believe on the back of this there will be a change to survival, but the results haven't been published yet, and it's too early to see that, but we're optimistic.
And then Shirley Wishart, apologise if I've said that wrong. Has there been any progress in research into replacement joints, orthosis or prosthetics?
Do you want me to start? So this is a really interesting area. There's been quite a surge in research in this area, and you might have heard of Stanmore implants, who have now been taken over by Stryker. So there's a huge amount of interest and effort into developing new implants, better implants that have got reduced rates of infection and various things. So it's a growing area of interest, and there's quite a lot of money being put into that. There's also an initiative to improve and increase surgical trials. So the National Institute for Health Research and IHR are hoping to stimulate more surgical trials, research trials. And some of our surgeons are interested in running those trials in bone sarcomas as well.
Yeah, I'll just add that also the design of the it's been linked now to the planning of the surgery. So advances in 3D printing, for example, they really facilitate an imaging and coordinated on how the surgeon actually can see the tumor and able to perform the surgery. And at that point, the designers of the implants are being brought on board and participating at the same time. So definitely the big improvement, 100%.
Yeah, and there's also improvements in prosthetics as well. So prosthetics are getting much more sophisticated, and actually the NHS prosthetics are getting better as well, and some of our oncologists are prescribing is probably the wrong word, but recommending that young people have access to kind of athletic prosthetics as well, which is really encouraging.
Good. Okay, so a question raised on the chat. Is there any current research into dedifferentiated chondrosarcoma?
There is. So actually it was a bit of a serendipitous kind of research finding. So it was a project that was funded looking into Ewing's and osteo in the first instance, and it was looking at the role of MMP in the development and the metastasis of these bone sarcomas. And actually, when the results were analysed, often researchers include cell lines from other bone sarcomas as well to compare. And actually, it was shown that it's more important in aggressive chondrosarcomas, and in particular, if chondrosarcoma. And that researcher has developed a really impressive model of dedifferentiated chondrosarcoma, which is hopefully going to use in further research.
Just the same person asked sort of the following question: how are the IDH inhibitor trials for chondrosarcoma going?
So at this point, well, so you might have noticed, Professor Flanagan is actually going to give one of the upcoming seminars. So she's obviously been doing this research. So I mean, we can give you some update, but I think it might be better if
we'll not steal Professor Flanagan's thunder.
Yeah, I mean, you're going to steal her thunder.
You need to tune back in, hear it from a horse's mouth, as it were. Okay.
Yeah, there's also trials going on at the Royal Marsden, and
True.
We heard some of a slight update in it was November, I think, at the sarcoma meeting, but the true results, the outcomes, we'll not know them for a little while yet until the data's been analysed.
Okay, so there was a question that was asked that caused a bit of a discussion. So I'm going to ask the original question anyway. Chris Hassel asked, have or would BCIT be able to tap into Foundation 1 testing based in the US?
Okay.
It might be
interesting, actually.
We're in touch with across the US, maybe, as well as a why.
Yeah. So I'm not sure whether everyone knows, and I didn't mention it in the sample collection slide, but sarcoma patients are going to be eligible for their whole genome sequencing as part of standard of care. So it keeps sliding the actual date of when that's going to be introduced, but hopefully in the not too distant future, with the intention that results from that will inform of any potential treatments that they might be eligible for or benefit from. I think it will take a while to get into the full system of analysing that and then informing and patients getting treatment, but that's the direction of travel, is for us to analyse that as a standard and then be able to give patients more personalised treatments.
Louise has asked, what do you think the next 5 to 10 years hold for bone cancer research or bone sarcoma research?
Do you want to answer, or maybe? Tricky one.
Go on.
I don't think. I think the impact of genomic analysis is going to be fundamental for us to understand the genes that are responsible for the development of the tumors and pinpoint exactly which patient has what mutation, so therefore it can be treated appropriately. For me, that's just a really important area that I think within the next few years, very few years really, because it is becoming a reality, and a reality for primary bone cancer patients is really where the impact would be really, really felt. I don't know what you think, Zoe.
Yeah, no, I agree, and I think more practically, I think that we've seen it in our community. I think we've got a smaller research and clinical community, but we've got a very engaged community, and I think researchers and clinicians are starting to understand that they need to work closely, and we're seeing that, and I think that will really accelerate findings and our understanding.
Just to answer a couple more questions that have come in. So, Shia, I think that's right, Shia. Bernard's asked, can we get a copy of the slides emailed to us? Yes, we will do that. We'll save it as a PDF and we can distribute that so you can access them afterwards. Okay, so Nick has a question. Please can, oh, where are we? Sorry, let me find it. Do you know about the plans that the NHS has got for whole genome sequencing for everyone?
So yeah, so that's what we talked about earlier. I'm not sure when it's going to come live. It was supposed to be the start of the year, but it's been delayed a couple of times. But yeah, it will become standard of care. So patients will be consented, a bit like 100,000 genomes, but it will be standard of care for sample to be analysed and whole genome sequencing done. Just don't know when it's going to start, but we're hoping that our sample collection grants will support that and make sure that our patients do benefit from it. Okay, so we've come to time now, so I'm afraid that's it for questions. But if you do have any other questions for us or for Zoe or Vicky, you can send them to the email address which you've got your invitation from for this webinar. As mentioned earlier, this is the first of our webinars. We hope you found it informative and helpful. If you do have any feedback, please do send it to us on email. The next one is next Friday, and that's all about understanding DNA and RNA with Dr. Darryl Green. We have a further 4 that are confirmed. We will send the invitation to you all in advance of each webinar. It will be a different webinar link each time, so if you do want to join, just keep an eye on your emails. If you have enjoyed it, we would just ask you, if you're on social media, it'd be good to have you back on social media using hashtag BiteSizeResearch. But also, if you do have any positive comments or any recommendations, send it to us on email. But we hope you found it informative, everyone, and thank you very much. Also, if you have any suggestions for any webinars that you would like in the future, any subjects that you are particularly interested, please also send that and we'll try our best.
Yes, and also, so we really try to give you like little highlights of research every now and then. So if any of you is particularly interested in any of special areas or anything that we mentioned, we can just get back to us and we can maybe produce more. Great. All right, everyone. Thank you ever so much. I'm going to end the meeting now, but we hope you've enjoyed it. Take care. Thank you. Thank you.
Thanks very much. Bye.
Thank you. Bye-bye.